It's a little after the hour, so shall we get started? David and Wayne,
Rock and roll.
Let's rock and roll. So we have over 70 attendees, almost 80 attendees, which is phenomenal. Thank you all for joining. I'm Dr. Audrey Fan. I'm an assistant professor in the Department of Neurology and Biomedical Engineering. I'm delighted to invite you to our 9th annual ADRC Research Symposium at UC Davis. The ADRC at UC Davis, for those of you who may not know, has received continuous funding from the NIA for over 30 years, and we pride ourselves in excellence in our clinical care and research, but even more than that, in training the next generation of AD researchers in community outreach and many more things to serve our diverse community of caregivers as well as patients. So we're delighted to have you join. We hope this symposium will serve multiple roles. One is to share some awesome science, and I'll introduce our keynote in just a sec, but also to introduce our research development grants, which our director, Dr. Charlie Carli, will go over as a mechanism to seed funding for exciting and innovative new projects. We'll also share some rapid fire pitches from younger researchers. So we have a full agenda, and let's just get started. So it is also my pleasure to introduce our keynote, Dr. Jack Wells. He is, as the title says, a Wellcome, Trust, and Royal Society Sir Henry Dale Research Fellow. He leads a group at the UCL Center for Advanced Biomedical Imaging. He and I have interacted multiple times through the MRI society called the International Society for Magnetic Resonance in Medicine. It's where we got to know each other a little bit and exchange cat photos. But we're delighted to have him share some of his new MRI non-invasive methods to understand blood-brain barrier as well as blood CSF fluid motion, which has a lot of implications in early detection of Alzheimer's disease. So with that, I will turn over the podium to Dr. Jack Wells. Thank you for joining us.
Thank you so much for the introduction, Audrey, and for the opportunity to come along and speak today. And hello to everyone listening. I hope you're all safe and well during these difficult times. I should say, by the way, please do feel free to interrupt at any stage to ask questions, or if anything is unclear, don't hesitate to let me know. So I'm going to begin with a bit of the background to this project, which was primarily motivated by Alzheimer's disease. And I'm sure, as you are all aware, this is defined by the presence of these two proteins, amyloid beta and tau. Can you see my laser pointer, Audrey? Is that coming through okay?
Yes, I see it, definitely.
Okay, that's great. And these proteins are known to accumulate many years before patients present with clinical symptoms such as memory loss. And although the precise sequence of pathological events that leads to clinical symptoms remains controversial, overall there's a consensus that amyloid beta is a likely initiating molecule of the condition. And there was an interesting study published in 2015 in Science where they metabolically labeled amyloid. And doing this, they gathered evidence that suggests that in late onset sporadic cases, amyloid accumulates not because of increased rates of production in the brain, but because of decreased rates of clearance. And this has focused the minds of the Alzheimer's disease research community to try to get a better handle on the contribution of different clearance mechanisms to the removal of amyloid from the brain. And broadly, these include local enzymatic degradation, clearance across the blood-brain barrier into the circulation, and removal into the cerebral spinal fluid. And broadly, the motivation for our imaging project is if we can develop sensitive and practical imaging techniques to monitor the health of the brain's clearance pathways, then we may be able to identify patients who have impaired clearance, at which point intervene therapeutically, try to stimulate clearance, delay the accumulation of amyloid, and ultimately try to delay or even prevent clinical symptoms occurring later in life. And at this point forward, I'm going to concentrate on CSF-mediated brain clearance. Now, if you were to look up CSF Physiology in a biology textbook, the description would be broadly consistent with the following schematic video. It would describe how CSF is secreted at the choroplexus. Try and get the video to play. Yeah, secreted at the choroplexus, this unique tissue in the lateral ventricles. The CSF then fills up the various ventricular compartments, flows down the aqueducts and cisterns, and eventually reaches the subarachnoid space that surrounds the entirety of the brain's outer surface. It's then absorbed finally back into the blood through these little nodules called the arachnoid villi. Now, what perhaps jumps out from this classical description of CSF physiology is it doesn't in any way allude to any interaction of the CSF with the brain tissue itself. And this notion was challenged principally by two scientists, Michael Neerdegaard and Jeffrey Iliff at the University of Rochester, originally in 2012. And they proposed that in fact, CSF rapidly interacts with the brain's interstitial fluid. And it does this by, in a rapid fashion that's physiologically important. And this is enabled by a particular network of plumbing in the brain that they have defined as the glymphatic system. And I'm just going to, I'm sure again, this is familiar to many of you, but I'm just going to broadly describe its basic components using this simple schematic video. So this hypothesized pathway begins with the inflow of CSF from the subarachnoid space into the brain tissue along para-arterial channels. We then have exchange of fluid into the tissue. So we've got astrocytes in beige and neurons in gray here. And this creates a convective flux that acts to remove waste products, including amyloid beta, that then leave the brain along paravenous channels. So the continual inflow and exchange of CSF with interstitial fluid facilitates homeostasis of the brain's internal environment. And a key aspect of this pathway is the presence of these tiny water channels known as aquaporin 4 that sit at the interface between the perivascular space and the brain tissue at the end feet of astrocytes. Now, just to go into a bit more detail about the particular experimental evidence that's informed this hypothesized pathway, this largely derives from invasive experiments in animal models, and typically traces are injected into the CSF or into the brain tissue, and then their distribution is mapped. So for example, here in the mouse brain, traces are injected into the CSF at this location called the cisterna magna, just caudal of the cerebellum. And half an hour later, they remove the mouse brain. And in the case of the small fluorescent red tracer, you can see that it was fairly evenly or fairly diffusely distributed about the brain. And immediately this data kind of challenges the notion that CSF has minimal interaction with the brain's interstitial space. Larger traces they found to be basically stuck in para-arterial channels, and this informed the para-arterial inflow component of their pathway. Conversely, when they injected traces directly into the brain tissue, they found these were then co-localized with paravenous spaces, informing the paravenous outflow component of their pathway. They've also managed to capture aspects of the system dynamically, in this case using 2-photon imaging. So here we have a surface vessel in the mouse brain. And again, they've injected traces into the CSF, and they saw that these became co-localized with paravascular channels, and some of the traces even followed the penetrating arterioles that dive down into the cortex. Now, this pathway really came to prominence, I think, following a high-profile publication in Science, where they looked at lymphatic function in the sleeping brain. And they found that when the mouse was asleep, they observed rapid infiltration of CSF traces into the brain tissue, shown in green here. Conversely, when the mouse was awake, the amount of inflow of the CSF into the cortex was significantly impaired. Similarly, when the mouse was anesthetized, in this case with ketamine xylazine, they observed rapid infiltration of CSF traces into the tissue. And again, this was markedly stunted when the mouse was awake. And to try and understand this observation, they measured the volume of the extracellular space in the sleeping and awake state using immunophoresis. And they measured a 60% expansion in extracellular volume in the sleeping brain. And they argued, therefore, that this reduces the resistance to the inflow of CSF when the mouse is asleep and enables the lymphatic system to operate efficiently. They surmised, therefore, that perhaps a critical functionality of sleep was to enable the lymphatic system to operate efficiently and remove waste products that accumulate during the day when the brain is working hard. And there's since been a number of review articles which have speculated on the link between lymphatic function and sleeping patterns in Alzheimer's patients. So there's clearly mounting evidence that CSF plays an important role to support brain homeostasis, but our understanding of the precise anatomy and function of lymphatic pathways in the human brain is limited by a lack of non-invasive techniques for assessment. So there are many unanswered questions as to exactly how CSF is acting to interact with the brain tissue, particularly in the human brain. And so the aim of my fellowship over the past 4 years or so has been trying to develop non-invasive MR techniques that can assess the action of the CSF to support healthy brain function. And this is where we come on to MRI of brain fluid movement. And there are 3 techniques that we've developed so far. The first targets the function of the blood CSF barrier. The second tries to measure the movement of fluid in the perivascular space, the gateway to the lymphatic system. And the third tries to assess the presence of these small water channels, aquaporin 4, at the level of the blood-brain barrier. And today, I'm primarily going to focus on blood CSF barrier because it's the most recently developed technique. And Audrey mentioned that she would be particularly interested to hear about this method. But if there's time, I'll briefly touch on techniques number 2 and 3. So I'm sure, as everyone is aware, our brains require constant diffusion by the peripheral circulation to sustain normal function. And there are 2 barriers or interfaces that separate the blood from the brain. The first is the blood-brain barrier, and this describes the microvessels that perfuse the parenchymal tissue. And the second is the blood CSF barrier, as I mentioned earlier. And the main locus of this is the chloroplexus in the ventricles. Now, the structure of these 2 barriers is very distinct. So the blood-brain barrier, for example, we have this tight endothelial layer that's very restrictive in what it allows to transfer from the blood into the brain. Conversely, the endothelial layer in the blood CSF barrier is fairly leaky or fenestrated, but we have this very metabolically active layer of epithelial cells that has many active transport processes and is very selective in what it allows to transfer from the blood into the CSF and eventually the brain. And given its unique structure and location, the blood CSF barrier performs unique functions in support of brain health. And these can be broadly divided into delivery, clearance, and CSF secretion. And there are certain cells and molecules and antibodies that can either exclusively or primarily only access the brain from the blood via the blood CSF barrier. And it's also been shown to be an important conduit of clearance from the brain for amyloid beta, for example, and other substances that may be detrimental if allowed to accumulate. And perhaps its most famous role is the secretion of CSF, and it has been described as a kind of giant sweat gland for the brain, secreting about 400 milliliters per day in the human brain. And its role as the primary site of CSF secretion helps drive delivery and clearance through volume transmission. And there's a video I found which shows what this structure looks like in vivo. This was during endoscopic surgery in the human brain, and this was to remove a cyst in the chloroplexus. And you can see it has this very kind of unique coral or sort of sponge-like structure. And this illustrates that it's very well designed to kind of interface the blood with the CSF. And you can see that it moves around a fair bit and it kind of beats in concert with the heartbeat. And it also has this quite distinctive pinky reddish color owing to its high blood volume and high perfusion. So you can see it's a very unique structure. So unsurprisingly, given these unique functions, its dysfunction has been implicated in the development of many common neurological conditions. And this includes age-related cognitive decline, specifically Alzheimer's disease, enlarged ventricles associated with hydrocephalus, for example, and multiple sclerosis. But again, our understanding of its precise role in these conditions is limited by a lack of non-invasive techniques for assessment. When we consider this to well-known techniques such as fMRI BOLD or ASL, these methods are sensitive to perfusion, and in a sense they provide non-invasive surrogate maps of the function of the blood-brain barrier to supply local tissue with oxygen and nutrients. But there's no analogous technique for the blood CSF barrier, and that's what we've been trying to develop so far. And why is this? Well, I think the principal challenge is due to partial volume effects. So ubiquitously, functional imaging techniques, they tend to require quite large coarse voxels, trying to detect quite small signals. And so, for example, if we think of ASL, this is an ASL image in the mouse brain. This is directly proportional to blood flow. And if we impose a functional voxel on what we're trying to measure, the chloroplexus here, you can see we've got some chloroplexus tissue, but we've also got partial voluming from the parenchymal tissue and from the CSF, meaning it's difficult to disentangle these factors. And if we think about more invasive microscopy techniques, again, these have been hampered by its quite inaccessible location in the deep brain compared to looking at surface vessels on the cortex, for example. Although there's been some really nice work recently from Maria Leiferman's lab where they've used two-photon imaging to image this structure. So how has choroplexus function been measured in the literature? Well, typically this has been achieved by using autoradiographical and microsphere approaches in large animal models. And interestingly, these studies always show that perfusion to the choroplexus is about 5 to 10 times greater than blood flow to the brain. So the approach that we've taken is by using an established technique called arterial spin labeling, and we call the new technique blood CSF barrier, ASL. And ASL, again, I'm sure it's a technique that's very familiar to many of you, but it uses water molecules in the blood as an endogenous tracer to map perfusion to the brain. And the way it works is we apply a labeling pulse that intersects the neck, and water molecules on their journey towards the brain, they become labeled. And we then wait a few seconds for the bolus of magnetically tagged water to arrive in our region of interest, and we acquire an image. And in the case of the mouse brain, we can then use this data to generate a quantitative map of perfusion in recognized physiological units. Now, with the new technique, all we do is we apply our labeling, but then when we acquire our image, we just turn the echo time up to 11, in this case, 220 milliseconds at 9.4 Tesla. And this means that both empirically, knowing the T2 of tissue and blood, and theoretically, knowing the T2 of tissue and blood, and empirically, when we acquire the image, we can quite unambiguously null the signal from tissue and blood, and we're left with an image that just contains MR signal from the CSF. And this means that in theory, when we do this experiment, we can isolate the signal from our labeled blood water that has been delivered to the CSF by the blood CSF barrier. And this means that we can overcome partial volume effects and that would derive from the surrounding blood and tissue, for example. And because this is an ASL technique, we can then fit this data to a model to actually quantify the total amount of blood CSF barrier mediated ventricular blood water delivery. And I hope that this measurement may represent a meaningful, non-invasive surrogate measurement of blood CSF barrier function. So if you were to do this experiment, just to give you a feel for what the data looks like that comes out. So again, this is in the mouse brain. These are our standard ASL image. This is the control, and this is just a standard subtracted image. We increase the echo time, and again, we just see signal from the lateral ventricles within our slice. And then when we look at the subtracted image, we can measure this positive coherent signal. The problem is it's a very small signal. And when we first did this, we were very skeptical that this may well be confounded by artifacts. So because it's such a tiny signal, it might reflect some imperfections of the labeling and control pulses on our imaging slice, or maybe reflecting local CSF movement in an analogous way to the time slip technique that's actually designed to look at CSF movement. So we performed a series of experiments to try to convince ourselves that this signal is genuine and does reflect this specific physiological process and this surrogate measure of blood CSF barrier function. And I've called these characterization and validation, and I'm just going to go through these. So firstly, we looked at the time course of this signal. And so firstly, looking at standard ASL, when you increase the inflow time or post-labeling delay time, you see this characteristic increase in signal as more labeled blood is delivered to our imaging slice, and then decrease because of the relatively short T1 decay of the tracer. With the new technique, just increasing the echo time, first of all, the lateral ventricles are lighting up as expected, and we see this delayed arrival, and this is because we need to wait longer now for our labeled blood water to cross the blood CSF barrier before it can be detected. We see also a reduced rate of T1 of decay because of the longer T1 of CSF. And this difference in the dynamic signal curve is very robust across the 12 mice that we looked at. Now, next, the choroplexus is situated in distinct locations within the ventricle system. So this is a segmented 3D view of the ventricles in the mouse brain from the Allen brain atlas. And the choroplexus is situated in the caudal aspect of the lateral ventricles and not the rostral. And so when we did our measurement at these two locations, we measured a marked increase in the caudal section. And this provides evidence that our measure is co-localized to the location of the choroplexus within the lateral ventricles. So we're beginning to build up some confidence in our measurement. Next, what we did is we deliberately modulated the labeling efficiency. So normally in ASL, you try to maximize your labeling efficiency to get good SNR. But in this case, we deliberately reduced it using techniques described in this quite boring paper that we published a few years ago. And doing this, we found that the effect of changing the labeling efficiency had a more or less identical effect on the established and validated standard cortical ASL signal with the new blood CSF barrier ASL signal. This is across 5 different mice. And this provides quite strong evidence, I think, that the source of the signal between them is shared, and it reflects labeled blood that has been delivered to the brain and not local CSF movement. The next thing we did was we applied a combined diffusion and ASL sequence using methods that we previously described, and such sequences have been used previously to look at the permeability of the brain's blood vessels to water. And when we looked at the standard ASL sequence, we found that the pseudo-diffusion coefficient of our labeled blood water was greater than the control signal, which is an approximation for the ADC of the tissue. And this was as expected because there is a contribution from our labeled blood water that's still in the vessels, which has a high pseudo-diffusion coefficient, similar to the IVIM literature. With our new blood CSF barrier ASL signal, the pseudo-diffusion coefficient of the ASL signal and of the control, the CSF, was very similar across 3 different inflow times. And this suggests that we're not seeing any vascular contamination, and the source of our signal does indeed derive from the labeled blood water that's been delivered to the CSF. And finally, there was a previous study published in 1990 where they measured choroplexus perfusion using microspheres in rabbits, and they observed that endogenous hormone vasopressin significantly decreased blood flow to the choroplexus without changing parenchymal perfusion. And we can now do similar experiments with our non-invasive technique where we can take a baseline measure, give a drug or vehicle, and measure the response. And so looking at standard blood-brain barrier ASL, there was no change in cortical perfusion with vehicle or vasopressin. But looking at the blood CSF barrier ASL signal, we observed a 63% decrease with vasopressin and none with vehicle. So again, this, I think, provides quite strong evidence for the specificity of the method as a correlate of blood CSF barrier function. So at this stage, I think although we were quite paranoid throughout that this was some sort of artifact of such a small signal, I think we were fairly confident that we were indeed measuring what we thought we were measuring. And what you're seeing here, so this is across the 12 plots represent 12 separate healthy adult mice, and the crosses just represent the blood CSF barrier ASL signal across the lateral ventricles. And the solid line represents an adapted 1-compartment Buxton model fit to the data. And you can see that the data fits the model fairly well. And again, this shows that despite the fact it's a small signal, we can actually reliably capture this. I think because the slice thickness and the voxel resolution is so coarse, this means that we are actually able to precisely measure this signal. And also it's quite encouraging that the model fits the data fairly well, because the model, after all, describes the delivery and exchange of a tracer to a kind of sink. So again, this does support the validity of our technique. So fitting the data to the model, we actually calculated the mean rate of water delivery from the blood to the CSF to be 24 mils per 100 mil of CSF per minute. And the problem is to try to convert this to more recognized physiological units of blood flow, which are expressed in mils per 100 grams per minute of choroplexus tissue. We needed to know the mass of the choroplexus within our lateral ventricles. So in the same mice that we imaged, we performed histology, and we sliced the brain across the lateral ventricles from rostral to caudal, seeing this kind of characteristic pattern in the area of the choroplexus across this region. We then converted to a volume, and making some assumptions about the density, we converted to a mass, which was fairly tight across the group. And knowing the mass and knowing the total amount of blood water delivery, we can then actually calculate the perfusion of the choroplexus tissue if we assume a 100% water extraction fraction. And we calculated this to be 1,265 mils per 100 gram per minute. And when we compare this to cortical blood flow quantified using standard ASL in the same mice, we can see that in terms of the magnitude of difference, it's roughly 5 times greater, which is broadly consistent with those previous studies. So it looks like, again, we're in the right ballpark for this measurement. So having convinced ourselves that this was working, we tried to think how we could use this method. And there's been a number of studies that have shown quite marked structural and functional changes to the choroplexus with aging. And so here in the rat brain, they observed quite clear morphological changes in the aged rat brain, in the aged choroplexus. In this study, they looked at the isolated perfused sheep choroplexus, and they measured a clear trend in decreased CSF secretion with aging. In this study, they measured CSF secretion in humans using an intraventricular catheter, and they measured a non-significant trend for a reduction in CSF secretion with aging. And very recently, Mike and Nedergaard's group, they've measured again about 30% reduction in CSF secretion in the aging mouse brain. So we applied our technique to 6 and 23-month-old mice. This corresponds to roughly 30 and 65 human years. And first of all, looking at more standard imaging biomarkers, there was no significant change in the volume of the lateral ventricles between the groups. There was a subtle decrease in cortical T1, although this is a fairly non-specific biomarker, and no significant change in the T1 of the CSF. When we looked at blood flow to the cortex using standard ASL, we measured a significant 15% decrease, and this is consistent with previous reports of cortical hypoperfusion in the aging brain. But promisingly, when we applied our new technique, we observed a 40% significant decrease in the rate of blood CSF barrier-mediated water delivery to the lateral ventricles. And this is quite encouraging because I believe that currently perfusion measurements using ASL are thought to be a leading candidate biomarker of early age-related neurodegenerative processes. So the fact that we can see quite a more marked, a greater effect size at this age is quite encouraging. And indeed, a few months ago, there was a paper that suggested that perhaps in humans looking at transcriptome data, they see the biggest change in the choroplexus in the human brain between the age of 50 and 60 in the human brain. So perhaps, again, we're reflecting this earlier transition in the choroplexus. And when we looked at the structure of the choroplexus in these mice, we saw visually that the changes were broadly consistent with more detailed characterization in previous studies. So the other thing we wanted to look at was whether we could use the technique to try and gain some insight into CSF secretion processes and the relationship of choroplexus function to lateral ventricle volume. And it's known that the size of the lateral ventricle volume, the lateral ventricles, reflects a balance between rates of CSF secretion and CSF absorption. And if this becomes imbalanced, then this can lead to enlarged ventricles, which is linked to hydrocephalus and neuropsychiatric disorders. And as we know, a major function of the blood CSF barrier is the secretion of CSF. And this is exemplified by the classical experiments of Dandy, where he dissected the choroplexus in the anesthetized dog, and he observed collapse of the corresponding lateral ventricle. And more recently, there's a very nice study where they looked at an experimental model of post-hemorrhagic subarachnoid hemorrhage, and they observed an inflammatory state of the choroplexus, which resulted in increased CSF secretion and subsequent enlargement of the lateral ventricles within a 24-hour period. So we wanted to try to understand whether our technique could give us a meaningful correlate of the action of the choroplexus to determine lateral ventricle volume. And to do this, we exploited the fact that in the healthy adult brain, there always appears to be a really marked range in the volume of the lateral ventricles. This appears to be also kind of ubiquitous across species. So here, for example, in the adult mouse brain, and lateral ventricle volume, remember, is very easy to precisely measure with MRI, but we see a really marked range. And this is, again, borne out in other species. So for example, a previous study in the rat brain, you can see this rat's got very small ventricles and this one much larger. And indeed, in the human brain, you can see that there's a huge range in the volume of the lateral ventricles in healthy young volunteers. So exploiting this, we hypothesized a positive correlation between our new measure of blood CSF barrier function and lateral ventricle volume. And just to give you an example of what the individual animal data looks like, we see that in adult mice that have relatively small ventricles, our new functional measure is much diminished in comparison to mice which have larger ventricles, where we measure rapid delivery of labeled blood water to the CSF. And this was borne out convincingly in 2 separate cohorts of adult mice, where we see a strong correlation between the volume of the lateral ventricles and the total amount of labile blood water delivery by the choroplexus. Interestingly, though, in our aged cohorts, so in 2 separate cohorts of aged mice, again, we see this global reduction in blood CSF barrier function, but we don't see this correlation. So there may be age-related changes in CSF absorption pathways that are at play here. So this does provide evidence that in the adult mouse brain, we can non-invasively capture the action of the blood CSF barrier to play a role in the determination of lateral ventricle volume, and provides circumstantial evidence that our measure may correlate to CSF secretion. So in support of this study, we performed a separate study where we took measurements of blood CSF barrier function in the same cohort at baseline and then 24 hours later, and we saw pretty good reproducibility, with the mean error being 1.3 mil per 100 grams per minute. And so this provides evidence that the range in blood CSF barrier functional measurements that we're detecting here are primarily physiological rather than methodological in origin. So finally, we've been working on human translation, and I've been seeking the help of my colleagues David Thomas and Magdalena Sokolska, who are much more experienced in human scanners than I am. And we've been hitting some problems with noisiness of the measurements and instability at such a long echo time. And so here we're seeing that overall we do see an increase in our signal where we look at regions in the CSF that are co-localized with the choroplexus in comparison to regions which are not, but we're looking to try to develop strategies to reduce the noise to really get this technique running in a clinically feasible scan time. So this is a work in progress still. So to conclude, this is the first non-invasive technique to assess blood CSF barrier function, and it works by quantifying the rate of blood CSF barrier mediated arterial blood water delivery to the lateral ventricles. This therefore reflects the mass of the choroplexus involved with its overall perfusion and the extraction fraction to water. Promisingly, we observed a 40% reduction in this measurement in the aged brain, and suggests that perhaps the choroplexus may be particularly vulnerable to the aging process. And I think it would be interesting to look at this measurement in the human brain in aging. And we observe a close correlation between ventricle size and blood CSF barrier function. And so I think, again, it would be really useful to use this imaging tool to try to better understand the etiology of conditions such as hydrocephalus, which are defined by large ventricles. And we're continuing to work on human translation, though there are some technical hurdles that we need to overcome. At this stage, Audrey, shall I take a break for questions, or shall I just carry on through to talk about some of the other techniques?
I think we could take a quick break, and I actually would like to just briefly give a shout out to Dr. Wayne Meyer and Dr. David Johnson, who are our co-hosts. And actually, Dr. Johnson is monitoring the questions. I do believe there were a few questions. So I wonder, Dr. Johnson, if you want to just take a quick break and take some of the questions so far. Yeah, let's do that.
You got it. I'll read some of the questions first in the order in which we received them. First, Sacha asked, Dr. Wells, could you briefly comment on the role of the ABC type drug efflux transporters in the elimination of amyloids and other waste products? Do they matter? And are they a key determinant of elimination in the blood-brain barrier for the BCSFB? For example, P glycoprotein.
So I must say that I'm an MRI physicist by training, and my understanding of the biology of the clearance mechanisms of the brain is not quite as good as it should be. And so I know that there are very important mechanisms, so the LiPo1 and RAGE, for example, that facilitate clearance across the blood-brain barrier, but I'm not quite up to speed on the latest drugs that target those mechanisms. But there's no doubt that there are a number of different clearance mechanisms which are currently being targeted and are promising therapeutic targets that people are looking at. But I'm sorry, I can't really give you a very informed answer.
No, I think that's still fair and exciting. This is an exciting area. Paul Miskowski, Dr. Paul Miskowski asked, how applicable or complicated would this technology be to evaluate movement of specific molecules across the blood CSF barrier? For example, large molecules like insulin or specific pharmacologic compounds?
I think so. This technique really exploits. It sort of really builds on similar principles to arterial spin labeling, in that we can take advantage of the abundance of water in the body, and therefore use standard MRI imaging to actually use it as a tracer. I think for looking at other molecules, to me, it isn't obvious how you'd have to think about maybe a spectroscopy approach, but I just don't think, because the SNR of ASL is very small anyway, and then superimposing the SNR challenges of spectroscopy, I think it would be quite challenging. So I think you might have to think about more nuclear imaging techniques for those sorts of studies.
Very fair. From Andred, the compliments you on a very nice talk. Are you looking to apply this model specifically to the AD mouse brain? All of your mice so far in your talk were healthy wild types, I believe. Are you using it in genetically modified mice like the AD mice?
So yeah, so we just got to the stage really where we've got it up and running, and there's all sorts of potential applications for this method, because in the literature, I think the measures of blood CSF barrier function have been really understudied. So there's huge potential to use this technique to look at different models, and that's definitely something that we're thinking about. And if other people would like to try and get this technique up and running and have ideas of how to apply it, I'd be more than happy to help in any way that I can. But yeah, so the study by Mike and Neerdegaard I mentioned earlier, they looked at an APP mouse model, and they observed I think about a 30% reduction in CSF secretion. So it'd be really interesting to see if we could replicate that kind of reduction in blood CSF barrier function using this technique.
Great, thank you. Dr. Carli asks, you suggest that your method measures blood CSF water exchange. Would it be more accurate to say that it measures choroid plexus blood exchange? Although seemingly a semantic difference, actually the choroid plexus is designed to exchange water, whereas the remainder of the blood-brain barrier may have a different physiology.
That's really interesting. So my understanding, and I might have got, because I'm not an anatomist, but I may have got some of the semantics slightly mixed up. But my understanding that the blood CSF barrier and the choroid plexus are terms that are used interchangeably, and so it may well have been better to call this a measurement of choroid plexus function along those lines. But I chose to use the term blood CSF barrier just because I thought it helped emphasize why looking at this particular functional tissue is important.
Yeah, I'll just step in here. I think this is fantastic. I don't want to be critical, but in the sense that as both a neurologist and a medical person who understands the physiology, really the choroid plexus is like a kidney. It really is a filter. It's designed, and you made a few comments in your picture show that this is it. You show cortex above in the little red, suggesting that this has implications about the cortex, how water shifts in the blood-brain barrier outside of the choroid plexus. And that was my comment, that I think that the blood-brain barrier at the microvascular outside of CSF in the tissue is different than the choroid plexus CSF barrier, which is designed to filter and create fluid for the brain to sort of work like a kidney. I think that your analogy about a sponge is really absolutely right on, and it has very similar characteristics to the kidney in physiology as well. So I think that's relevant, and I think it's really cool to be able to look at this to say, is this going up or down? And I suspect in certain diseases, you'll be able to, once you get the human stuff sorted out, and even in the AD mice, you'll be able to see that it's probably not changed too much in the AD mice, because it's just filling a bigger spot that's what we call ex vacuo. But that's a really cool thing because it may be changed in other conditions where there's an increase.
Yeah, no, thank you. Yeah, thank you for your comments. That makes sense. And particularly, I think in hydrocephalus is where I maybe see the application of this technique, just because there are treatments available for hydrocephalus at the moment, and so if we can use this to guide those treatments and maybe reduce the need for shunt surgery, that would be fantastic.
Excellent work.
We have a few more questions from the audience. Dr. Fan, do we have a few more minutes?
Yeah, let's take just a few more minutes and then give Jack the 10 more minutes to add a bonus.
We have 3 more standing. Min Zhao asks you, Dr. Wells, in addition to the flow of water, have you considered electrolytes and how they may be passing?
So again, I focused on water delivery just because that's something that we could measure with MRI, and this measurement will, I think, therefore, as I mentioned, reflect the fusion of the choroplexus involved with its mass and the extraction fraction to water, and how that measure would reflect changes in electrolytes. Perhaps that would be related to metabolism, which might correlate to perfusion. But again, that's something that would be interesting to investigate by comparing this technique with more invasive measures of those properties of the choroplexus. And that's something that we'd like to do. We'd like to work with others to do if they were interested. But again, I'm sorry, I don't have quite the answer, I'm afraid.
Sounds like an invitation to Dr. Zhao. Hangzheng Zhang asks, instead of following water, what about using 2-deoxyglucose, an old-fashioned tool of measuring CSF volume and movement? So do you have a comment for old techniques too, or?
Yes. So that, I think that would be interesting. Whether that technique is more relevant for looking at CSF movement, and whether it's possible to use those measurements to look specifically at choroplexus function, I'm not sure. Again, because I think partial volume effects are going to be problematic in FDG PET data with the fairly large voxels. I think it's going to be hard to distinguish the perfusion of the surrounding parenchymal tissue with this complex ventricular structure when you've imposed your slices and voxels on that from the specific metabolism of the choroplexus. But yeah, I'd like to do more reading on that literature, because there might be some relationship there.
I'm actually really curious about Nina Foltz's question.
That's next.
Oh, yep.
Well, go ahead and ask it, Auntie.
Sorry. So, for this technique, especially for humans, could you apply it to the 4th ventricle to reduce the amount of time for the ASL tag as compared to the lateral ventricles, like for the tag to arrive to the ventricles, I think?
Yeah, so in principle, it should work exactly the same on the 4th ventricle or the 3rd ventricle where there is choroplexus tissue, and maybe having a reduced arterial transit time would help the SNR. Though I think the motivation for looking at the lateral ventricles was, I think the majority of choroplexus is located there, in addition to the kind of ease of shimming and imaging that structure, where in comparison to looking somewhere a bit further down the brain near the brainstem. So I think probably I would suggest starting with the lateral ventricles because there's lots of choroplexus there. But in time, it should be easy because this is a single slice approach, but ASL should be easy to do whole brain. So we'd like to kind of extend it to that fairly soon.
Well, that is the last question that I'm tracking in the chats and the question and answer section. If there are attendees that would like to raise your hand and ask a question right now, we invite you to do so. We're searching for hands raised, and I am not seeing any, so I think it's time to thank you, Dr. Wells.
Yeah, would you like to, we have 12 more minutes, and I know you had a little bit of bonus material to share. If you have, 10 minutes of other methods, I think we'd all love to see it.
Sure, I'd be delighted to carry on for a bit. If anyone's, bored at me just rabbiting on, then feel free to just sign off. I will carry on for a bit longer. So the second technique I'm going to talk about.
Don't you dare! We have more things to talk about.
Okay.
Everybody, stay on.
That sounds good. So the second technique is looking at the perivascular fluid movement, which is a bit more directly related to the lymphatic system that I described in the introduction. And again, I just thought I'd share this video because I think it just provides such a beautiful depiction of what we're trying to look at here. And this is from a paper by Douglas Kelly's group, Mike and Neerdegaard. And again, they've got 2-photon imaging. You've got a vessel on the surface of the mouse, and they've got fluorescent particles in the CSF, and they performed particle tracking to look at the trajectory of these particles. And again, they're situated in this location that they call the para-arterial space, just surrounding and perpendicular to the orientation of the vessel. And you can see that they are propelled forward with a kind of coherent bulk flow type fluid movement, and they appear to be driven by the pulsation of the vessel. And I think this is just such a beautiful description because for years, for example, I was using MRI to measure blood flow to the brain, completely naive to the fact that there is this parallel circulation, which although flows at a rate of about, or velocity of about 1,000 times smaller, nonetheless does appear to be physiologically important. So the question is, can we capture this subtle physiological process using non-invasive MR techniques? And the first thing was to try to understand what the size of this compartment was. And it was quite difficult to determine that initially, but it was important to see whether we had any hope of actually spatially resolving this compartment using non-invasive MR methods. And what gave us some encouragement was a paper by Robert Thorne's group, and they delivered fluorescent traces to the CNS or the rat brain non-invasively through intranasal administration. They then took out the brain, and half an hour later, they saw that these traces were co-localized to paravascular spaces. And again, I had no idea that there was this fairly direct connection between the nose and the brain. This appeared to be mediated by processes similar to the lymphatic system. But they showed in this work that the size of the perivascular space around the large vessels, the large arterial vessels on the outer surface of the rat brain was kind of appreciable to that of the vessel itself. And so we thought this would be a good place to start in terms of actually trying to image this compartment. And given that it's, again, meant to be filled with CSF, we used a fast spin echo T2-weighted high resolution approach. And we looked at a lot of the feeding vessels around the outer surface of the rat brain. And where we saw an anatomy or a morphology that appeared to be consistent with our understanding of the para-arterial space was around the middle cerebral artery at the ventral aspect. And again, we've applied a very long echo time, and we're seeing bright signal from the fluid-filled compartments, the subarachnoid space at the base, the third ventricle, and these bright tracks either side of the vessel, which we think are representative of the perivascular space. And having acquired this image, we wanted to compare it with more established methods of looking at lymphatic function. So we used dynamic contrast enhanced measurements. We injected gadolinium into the cisterna magna, and then we took 2 T1 weighted images at time points after. And at baseline, again, you can see a bright signal from the vessel. And 20 minutes later, we can see evidence for a kind of radial inflow of gadolinium into the tissue, centering around these putative perivascular spaces. So this seemed like a sensible target for a non-invasive measure of the CSF-ISF exchange function. And we then tried to think if there was a way that we could actually get a readout of perivascular fluid movement. And this brings me on to one of several controversies about the lymphatic system. And this concerns the nature of fluid movement within this compartment. Now, the lymphatics guys quite strongly argue that there is coherent bulk flow from the para-arterial space through the tissue and into the paravenous space. And looking at the video that I showed earlier, it's sort of easy to understand where they're coming from. But there have been a number of papers that have suggested that perhaps what is more likely occurring is this idea of rapid pulsatile-driven dispersion. And this could act to drive fluorescent contrast agents down a concentration gradient that would kind of mimic the appearance of bulk flow, but actually is more of a dispersive mechanism. Now, given this uncertainty, we thought a good place to start to try to look at this fluid movement was a diffusion MRI approach. And this is because it has established sensitivity to this kind of dispersion with zero net flow effect. But we've also shown from some work we published a few years ago that diffusion sequences, if appropriately tuned, can be sensitive to more bulk flow processes, in this case looking at the major vessels in the rat brain. So we thought, given this uncertainty, this would be a good place to start. And to try to examine whether our approach was sensitive to perivascular fluid movement, we applied our motion probe gradients in different directions in a very similar way to what is done in diffusion tractography. And our hypothesis was that we'd see a greater pseudo diffusion coefficient when our motion probe gradient was parallel to the known direction of fluid within this compartment. And we saw this quite convincingly across, in this case, 10 rats that we looked at. And what you're seeing here, I'm sorry if I'm going to do this quite quickly, but what you're seeing here is when we apply our diffusion or motion probe gradient parallel to the orientation of these vessels, the pseudo diffusion coefficient is significantly greater than when it's applied perpendicular. We then extended this to a tensor, and again we saw that the principal direction of these tensors in the right and left perivascular space was parallel to the known direction of flow. And this provides some evidence that we are sensitive to fluid movement in this compartment. And you can extend this to map a tensor map of fluid movement. And we showed also that this measurement was significantly reduced in the dead brain and became isotropic as expected. Now, given previous evidence from 2-photon imaging that the pulsation of the vessels is an important driving mechanisms for perivascular flow, we gated our acquisition to the heartbeat, and we acquired an image during vascular pulsation 25 milliseconds after the R wave. And doing this, we saw that our signal was significantly attenuated in a very directionally orientated manner during vascular pulsation. And conversely, when we acquired during systole, when the vessel wasn't pulsating, that the extent of attenuation was markedly reduced. And I'll just dwell on this slide for a second, because I think this gets at the heart of the method, in that the limitation of the technique is it doesn't tell us anything about more coherent bulk flow versus rapid dispersion, but it does have very high directional sensitivity, and it's very sensitive to the pulsation of the vessels, which I think could be interesting. And we can then use this to generate a map of the pseudo diffusion coefficient. And we saw that this compartment really lights up when the vessel is pulsating in a very directionally oriented manner. And this was robust across the 5 rats that we looked at. And sure enough, when we increased pulsation giving G-butamine, we measured a significantly increased pseudo diffusion coefficient within this perivascular space. More recently, my student Phoebe Evans, she's been looking at taking these measurements over multiple delays from the ECG wave, looking at the effect of vascular pulsation across the heartbeat. And we see this quite nice characteristic curve that is present in the perivascular space, but not so much in the third ventricle. And so I think it would be interesting to use this technique to look at the relationship between vascular pulsation and age-associated pathology due to hypertension or atherosclerosis, and how this interacts with this measurement of perivascular fluid movement. So I'm sorry if I went over this in a bit fairly rushed manner, but just briefly, this is a new technique to look at the movement of fluid in the perivascular space. And this is an important conduit that helps to mediate the exchange of cerebral spinal fluid with the brain's interstitial fluid, which appears to be important for the removal of amyloid beta. And again, using an ultra-long echo time, we can help to reduce the signal from blood and tissue and get a signal that largely contains the fluid-filled spaces. In the directionality of our measurements provides some evidence for sensitivity to the movement of this compartment, and it's very clearly driven by cardiac pulsatility. And those who are interested can read more about this technique in our recent publication. How long have I got, Audrey?
I think if you can wrap up in the next couple of minutes and just see if anyone has any final burning questions, that'd be great.
Okay, that sounds good. I might have time just very briefly to touch on technique number 3, and this was led by Yolanda Ohini, who's now taken a postdoc in Laura Parks's group. And this was using techniques which are used both preclinically and clinically, and they combine arterial spin labeling with other contrast mechanisms such as diffusion, or in this case, multiple echo times, to try to measure the permeability of the blood-brain barrier to water. And our hypothesis was that this permeability should be very sensitive to the presence of these aquaporin 4 channels at the level of the blood-brain barrier. And so we did this measurement in a wild type mouse and in a aquaporin-4 knockout mouse. And when we looked at more standard MR biomarkers such as ADC, blood flow, and arterial transit time, we saw no difference between the groups. But as we hypothesized, we saw a greater exchange time, which is this quantitative surrogate measure of blood-brain barrier permeability in the aquaporin-4 knockout mouse, suggesting that we're able to capture the removal of these water channels that help mediate rapid exchange between water molecules in the blood and into the brain tissue. And so I think it would be interesting, again, to use these type of techniques to try to understand subtle changes in water permeability that may predispose more gross changes in blood-brain barrier structure that might be detected using gadolinium-based methods, for example. So that was very, very brief, but I'll just draw it all to a close by thanking members of my group, Yolanda, Phoebe, and Charith, and thanking in particular David Thomas, who is my PhD supervisor, who all these ideas really stem from many years ago. And finally, I'm based at UCL Centre for Advanced Biomedical Imaging, which was established and directed by Mark Lithgow, which is a great place to work. We've got all these great preclinical imaging techniques. And when things are better, if anyone would like to pop by and have a chat and have a tour, then that would be great. So do drop me a line. And thanks again, everyone, for listening. And let me know if you have any more questions. And sorry that last bit was a bit rushed.
No, thank you for sharing, and thank you for your awesome presentation. I do hope we can visit after things are a little better. I believe we have some questions, Dr. Johnson, if you want to.
Yes, I'm tracking one new question from Dr. Carli. Fantastic data. Did you measure blood pressure and velocity? We believe blood velocity may have a very strong effect on water diffusion out of the microvasculature.
Well, that's really interesting. So in terms of blood pressure, we've actually been, because in the Maestra and Douglas Kelly paper that I showed at the start, where you could see the particle tracking of perivascular flow, they actually increased blood pressure, and this showed that this reduced pulsatility and reduced the movement of perivascular fluid movement. And we're performing similar experiments, again using a pharmacological model of hypertension induced by angiotensin. And we are seeing some differences in these parameters. And we're trying to extend this by looking at an SHR chronic model of hypertension. So we're actively exploring similar angles. In terms of, I'm sorry, what was the other? Was it fluid movement, or I'm sorry, what was the other parameter you mentioned?
The second piece of this was that we believe blood velocity may have a strong effect on water diffusion out of the microvasculature. So have you been tracking the blood pressure and velocity?
So that's not something, so we've for quite a number of years, we've been using MR techniques to look at water movement out of the microvasculature and exchange. But interestingly, I've never come across looking at how that relates to blood velocity and blood pressure. I think there have been some studies by Alfonso Silva's group where they've shown that the greater the blood flow, and therefore perhaps by extension the greater the blood velocity, the reduction in the extraction of water, because I think there is less opportunity for exchange at the capillary bed. So it's interesting that that there might be a different relationship, but it's a very active area of research, exchange of water across the blood-brain barrier. So I'd be interested to learn more about that.
Excellent. So far, I see no more questions, but many accolades, Dr. Wells. The audience is loving this, so I wanted to give you that feedback.
Oh, thanks so much. That's wonderful, and thanks all for listening. And if anyone would like to get in touch, would like to collaborate, let me know.
Awesome. Well, thank you so much. You're welcome to join for the rest of our symposium, but I know it's quite late there, so we thank you for the time that you've shared. Right now, we'll take a bio break, and promptly at 10:10 here, we will resume with Dr. Carli's talk about our ADRC and the development research proposals. So let's take a quick break and promptly restart at 10:10. Thank you again, Jack Wells. All right, it is 10:10, so welcome back, everyone. This is the second half of our UC Davis ADRC 9th Annual Research Symposium. And I have the distinct pleasure of now introducing our UC Davis ADRC Director, Dr. Charles DeCarli. Charlie is our fearless leader of the ADRC, but he's also worn many hats related to mentorship, administrative leadership within neurology, and he also directs the IDEA Lab, which has served a core role in neuroimaging analysis and pipelines, as well as advanced methods related to Alzheimer's disease and cognitive decline. One of many accolades that Dr. DeCarli has received is quite recent, and he has received the largest grant to UC Davis Health ever. Was awarded to study across multiple sites white matter hyperintensities, its heterogeneous presentation in diverse populations, and it valued over nearly $54 million. So just an incredible career. So we're very lucky and proud to have you as our director. What Dr. DeCarli will do now is give you an overview of our ADRC in terms of our activities, who we are, but also to share the new format of our research development proposals, which will take the place of what was previously called pilot grants. So as Charlie is speaking, I'll type in a link that links to the RFA currently out. The applications will be due in early April. And with that, I'll let you take it away, Charlie.
Thanks, appreciate it. And thank you all for attending. I mean, this is an incredible turnout. It's fantastic. And we had such a wonderful lead-off speaker that just realizes how active this area of research is. I want to start by showing you just some samples of the people who have worked or are working at the UC Davis Alzheimer's Disease Research Center. And remind all of you that it really does take village to make this work. And in fact, ours is growing into a small city. So there are lots of people, and some of which are not on this slide, who make daily conscious success of the center. And I'm so grateful for all their help. Who are we? We're an interdisciplinary group of dedicated researchers, staff, administrators, and support staff are here to carry out the mission. We're really trying to understand Alzheimer's disease and related dementias through comprehensive patient evaluations and using research.
Dr. DeCarli, your microphone is cutting in and out. So if you could move closer to the microphone.
Okay, how about this? Is that better? Maybe I have a lousy connection.
That seems a little better to me. I was just going to comment on the same thing. Could you try?
Can you hear me now?
Yes, I think that is better.
I'll turn it up actually. So you'll see in the stack that cover a broad range of research. The important point of this talk is to see what we do and then lead into our new program and hopefully do to apply.
Charlie, I'm sorry, the audio is still not clear. Is there any way you can maybe switch it or just disconnect and reconnect to the audio?
Yeah, let me try. Let me turn this off and see if I can use the other audio. It's telling me my internet connection is unstable. Maybe the problem. All right. Let me close a bunch of stuff. Maybe that will help. You hear me now? Is that better?
Yes, that is better.
Oh, good.
One of the suggestions is maybe even to call in by audio or by phone if we continue to have problems, but this is better.
If you can hear me now, then I'm good.
Yes, we can hear you.
Okay, good. Just to share with you, we have two locations, two major clinical locations, but we're spread across the university. But we do look to recruiting participants both in Sacramento and in Walnut Creek. That is part in due part to the fact that we're trying to get diversity, and so we tend to go to geographic areas where we can find diversity. The Central Valley, of course, being Hispanic, Latino, and the East being more African American. We have a guiding conceptual model, and I think this is important. We're applying for our grant, our pilot grants, or now called Developmental Grant Program. And how it works is that we have a diverse participant study group that we are deeply phenotyping, focusing on risk and factors that include both biological diversity, which is genetics, vascular, and other diseases, and diverse life experiences. Then we look at various types of outcome measures that include brain integrity measures, pathology, vascular injury, et cetera, as well as clinical outcomes. And what primary goal here is to understand that emphasizing heterogeneity enhances the likelihood of discovery. So we try to get a broad clinical cohort that varies from essentially no education to a great deal of education across groups of diverse ancestral background, diverse pathologies, and then carefully evaluate them. And we have some themes that we focus on, and our major theme is to characterize the heterogeneity of cognitive aging and the factors that lead to the transition to mild cognitive impairment and dementia. Again, we're looking at risk and protective factors, genetic, psychosocial, etc. A broad range. We also have introduced recently plasma biological markers, and I'll talk about them in a little bit. We also have mixed pathologies. I believe, and our center has focused on this idea that aging and dementia are complex phenomenon. And to really be impactful is to develop a precision model approach or precision various components of these pathologies.
Charlie, I'm sorry, your audio has become garbled a little again. Do you think it's possible to call?
I know exactly what's happening. My wife is printing on the internet. Is it better now?
We can keep trying. I think sometimes it.
All right. Well, if you give me a phone number, I'll call in.
Yes, that might be better. Thank you.
What's the phone number?
Let me find that for you.
I just put it in the chat for you, Dr. DeCarli.
Thank you, Wayne. And sorry for the temporary interruption. We will be back.
I got it. Okay, I'm muted. Now you unmute me. That will work. Can you hear me?
Yes, I can hear you. Do you know which, so you're a phone in now, probably without a name.
Yeah.
What would
Hear
the
me?
area code be?
Pardon? 530?
Yes, I see it. Okay, so I think you are able to talk. Is that?
Can you hear me?
Yes.
Let's mute his video and see if he can talk through the phone.
Yeah.
Oops. Could you try speaking through the phone now?
Okay, can you hear me now?
Yes, that's my
Perfect.
Okay, all right. So getting back to this because of my at-home office being multitasking, I apologize, folks. So I'm on the updated themes, and our primary theme is to characterize the heterogeneity of cognitive aging. And we're going to do this a bunch of different ways using assessing clinical risk and protective factors, plasma biological markers, looking and examining mixed pathology, and then translating this into investigator-initiated interventions, which translated into English is basically our homegrown clinical trials. I won't be able to talk to you about them today, but I will say that we have some exciting stuff that comes from the basic science that was derived with these studies. And then we are working on caregiving as an emerging theme in our ADRC. One of the things that's happening in this period of the Alzheimer's Disease Center is that we're beginning to emerge as a research innovator. What do we mean by that? Really to help our expertise and leadership to innovate new study designs, novel methods, and to work as a testing lab for discovery that then will upscale into community-based studies. And in particular, one of the aspects that we have focused on is harmonization across studies such that we have very large data sets that can be used for research exploration. And this in part relates to the fact that Rachel Whitmer joined the group about 3 years ago and has a focus on population science and has brought in 3 very large community-based studies to help us with this endeavor. We also believe that by doing deep phenotyping of our participants, we can lead to testable questions, and that through creating resources for you all to use, you can help us in testing those questions or asking unique questions. And the final enhanced function is to use our basic science resources to help in clinical science translational activity development. We have grown to be a somewhat large center. I would say in the past we've been a sort of smaller to medium-sized center. We are now a fairly large center. We have about 118 staff and faculty. We have now have 7 cores and what's called a research education component. Many of you will be familiar with that if you've been participating with our research 2 times a month, our research meetings, and then our CPC, but also other activities that occur through the research education component. Our approach is really to use our clinical diversity science and our basic science to support creations of methods that are translated into population-based studies or discoveries that we hope would lead to therapeutic interventions, and that these then feed back in a positive feedback loop to inform our research objectives, and that these scientific advancements then will translate into overall concept of healthy brain aging. This is how we look at how we support you all and you're doing your interesting science, and that we support across the spectrum of science from basic to implementation. And implementation science goes beyond just clinical trials, but caregiver intervention, etc., psychosocial intervention. We have a relatively modest cohort that is very diverse. It's about 425 individuals. And you can see here that we span African American, Hispanic, and whites. And it's about 25%, and 50%. We do mostly mostly community-based research. So these are mostly from the community, which gives them representation. We have a spectrum of cognitive ability in the group that we follow. And the beauty of this is that we actually we're able to follow people transition across all of these syndromes from normal to dementia. And we have about 100 or so individuals that we followed longitudinally who have made that transition. This is just some of the clinical data we have. I want to draw your attention to, we have cognitive data, we have functional data, we have physical and motor, we have morphometrics, we have many questionnaires that include early life variables, interpersonal resilience factors, social, contextual, health behaviors. We also have disease-specific questionnaires that relate to vascular disease, medical comorbidity, and as you'll see, we have a great deal of biomarkers, and we are building up a caregiver information database that will, we hope, will lead to expanding caregiver research. Neuropathological. So I'm just going to walk through a couple of the resources that we have in our neuropath core. We have 546 frozen sample cases. We have paraffin blocks of over 900 cases. We have postmortem CSF, and we are creating an entire comprehensive digital microscopic slide library that Dr. Duggar has begun to use to advance into digital pathology and quantitative pathology. And here's an example of one of her publications showing the difference in the absolute estimation of amyloid versus our clinical estimations of amyloid. And you can see that it's a little hard to tell, but it's almost 100-fold difference between this sort of qualitative scale of no amyloid, sparse, moderate, and severe. And that this is an important step forward in understanding the complexities of and the multifactorial contributions of pathology to dementia. And Liwei Jin has been working very hard on interacting with our basic science colleagues through these, what's going to be now called the developmental projects. And the idea is that you all come to us with your interests, and we work in this basic science level using tissue, histopathology, rodent models, and other samples that may be available that then allows you to be successful researchers. And this is, I think, a unique characteristic of our center. We've just, with our recent reapplication or new application for an ADRC grant, we've begun to process fluid biomarkers, focusing on what's called the Alzheimer's ATN markers, which is amyloid, tau, and neurodegeneration is measured by neurofilament light. And we're using femtomolar sensitive methods called Simoa technology. And this allows us to do an in-depth assessment, and we've already started to see some really interesting findings coming from this. The biospecimens core resources consist of a unique biorepository from our longitudinal cohort, as well as any affiliated programs. Then we have not only, right now we have standard Simoa assessments, but we're also developing collaborations to get unique omics platforms that include all of these above. And then this is integrated with all the other data, and we're able to share that with you all. And just to give you a sense of what's available, so we have thousands of samples on plasma, serum, DNA, and RNA, which just enables a whole panoply of investigation. We also have a neuroimaging core that has its own great resources, and in its purpose is to support image acquisition, support processing, provide summary data, provide training, and then also to maintain our system-wide resources. And the goal here is to not only have pursue the goals of the ADRC, but also to create collaborations. And this is a summary slide, which I hope you'll take the opportunity to look at once you get the slide deck about what kind of resources we can provide. We're also moving into deep learning or convolutional neural net, and we found that this is turning out to be a quite satisfactory new area for us, and therefore we'll be generating even more extensive data from the neuroimaging analysis core. How do we share this data? One of the things that we're doing is involving tracking all of our specimens to be sure that we know where they are and how to share them and what components you might have available to you if you were to request them. And this is done through this open specimen method, and will be made more transparent to you all once you have requests. What we are moving to is a platform that will enable individuals to go to what we call a data sharing portal. We've created a new ADRC database. In this data sharing portal, there will be loads of information that will summarize what we have in the center, and then enable you to make requests based on what you see there and get clarification, and then follow through in any collaboration. The most important thing, however, for me to share with you is this new development that's going to occur in the Alzheimer's Disease Research Center. And as, in fact, in response to some of the feedback that we've gotten from you all who've had pilot project grants in the past, we are revamping them, and we are renaming them, calling them development projects. And hopefully this will be put in the chat, but it will also be on the in this slide deck, and you can click on it. But basically, it's a PDF that goes over all the instructions about the development project program. Our goal is to encourage new investigators or established investigators who are moving into a focus of Alzheimer's disease and related disorders. What is different is that we are funding a total, we're expecting to spend a total of $100,000 a year, every year of this next 5 years. And they will be focused on 1 to 3 developmental grants that can cover any limit of, or anywhere along the spectrum of resources that we have available. But what is really different is not only is the amount overall greater than what we've done in the past, but many of you who have had pilot project grants with us before have been concerned about such a short time limit of 1 year. This is now going to go away and will be up to 1 to 3 years in length, depending on what the needs of the application will be. As a consequence, there is going to be a slightly different approach, and that is we're going to have a multi-tiered funding approach, and it's going to be designed to optimize the utility of how this money is spent over a 3-year period. And we will, assess the various applications, but you all will have more flexibility, and I'll get to that in just an example. So, for example, then we will supervise this, and we will work more closely to each of the applicants. And we might actually even be able to expand or contract depending on the success of the application. For example, there probably will be greater funding for some projects that already have preliminary data showing promise, and they just need like an R21 kind of level of funding. Conversely, there might be somebody who has a really clever idea, but it's a high risk. We can give them a small seed amount, see what it looks like to get them, and then possibly extend that funding or expand that funding as their success emerges. What this will require is a developmental projects panel, and that's going to be a group that will meet regularly to assess how this is going. And we will then advise and support each of the applicants as they move through this experience. How will we evaluate these applications? It will be the applicant's qualifications. We do want to emphasize we're looking to develop junior faculty. I think we see that as our key mission. But we want novel and important science. We want technically sound methods. We want a strong study design. And of course, our statistics core will help people with questions about statistical veracity. We also want to be sure that the study design has in it aims that are attainable. Sometimes I find that some of these proposals can be overly ambitious. We will look for those who align with our areas of emphasis and make appropriate use of our resources. But finally, also that they have, we'll be looking for people, whether it's a novel idea and likelihood of further funding. How will this work? We'll have yearly call for applications. We're running behind this year simply because it has to do with, we move from one grant that we've had for 30 years to a new granting mechanism. And so our grant number will change. And until we know about our grant recipient, the receipt of our grant, we got pushed off. But in the starting later in 2021, we will start out by making announcements beginning in November. We'll look for, you can submit a one-page pre-proposal if you're not sure. And in fact, I've got emails where somebody's like, does this fit? That's the perfect thing for just a one-page pre-proposal. We encourage consultation with our ADC faculty. We encourage presentation at our weekly Adrc research meetings. And then if you want to do a full application, that's going to be a 5-page NIH type application. We, to be sure, we try to get the best people we can. We often invite 5 or more reviewers, either internally and externally, often a combination of both. We use the typical NIH scoring, but we give you feedback on all application. And some that are higher scoring but not quite meeting the threshold, we will engage in consultation and encourage resubmission. After the assessment, this is going to be a little bit different than the prior award. We're going to assign awardees to one or more of our executive committee members as liaisons, which is something we've always done. But we're going to be focusing on ongoing mentoring if the awardee is a junior faculty member. There will be a signed letter of commitment, and where we'll ask you to specify the expectation of the award and give us specific milestones. These can be adjusted, because what we will be doing is assessing these every 6 months with the idea that if you're moving along really well, we may expand the award. If you finish something and you're going on to an application for an R, et cetera, we can support that and close out the award. So I know that was kind of fast, and I apologize with the technical difficulties, but I'm happy to answer any questions at this point, and I'm going to stop my share. Maybe that will help.
Thank you, Charlie. We are just at time, and so maybe let's just take 2 or 3 minutes for questions. If anyone has any, feel free to put it in Q&A or chat, and I'll monitor the time so that we can let our pilot awardees present in a timely fashion. Are there any questions?
I'm reading this, and so I can answer Angela Zivkovic about interest in adding nutritional and physical activities. And the answer is yes, we are doing that. Yeah, Charlie, turn off your video. Okay, that seems to be it. But
yeah, if any questions come up, especially as you're looking,
yes. Can previous pilot awardees apply again? We discourage that, Dr. Olson, and because we want to give opportunity, but we can talk about other mechanisms by which previous pilot awardees can get involved in the center in a collaborative way to expand their research. Great, I'll hang up now then. Thank you all, and I apologize again. I really appreciate your patience. Take care.
The link to the RFA is in the chat, and I believe Charlie is going to stay on for the next presentation. So
there is one question that we just got in the question and answer section for Dr. Carli or whomever. We'll see who can answer this. Are there active collaborations with UCD Davis core facilities which analyze DNA and RNA contents of your plasma biomarker samples? I think Dr. Carli or Li Wei Chen or Dr. Duggar might answer that.
Yeah, you might not be able to hear me too well, but I'm back answering through my computer, but the answer is yes. And we will be happy to support that. And in fact, we're hiring new faculty with a genetic emphasis. So we're looking forward to those kind of collaborations.
Great. With that, again, please continue to put in questions in the chat for the next 10-15 minutes if you do have any. I would like to switch gears now and actually show some excellent examples of successful pilot awardees through the ADRC. And the first speaker, and that's quite alluded to already, but Dr. Pamela Line. She's a professor in the Department of Molecular Biosciences and was a pilot awardee in 2017 to 2018. Her lab develops new therapeutics to prevent brain damage after exposure to chemicals, and also understands how environmental factors intersect with genetic predispositions in neurodevelopment. So if you're able to share your screen, we're very excited for your talk.
Yeah, before I get going, I just want to mention one thing as a follow-up to David's wonderful presentation. Always exciting to hear David speak. And that is that the triculture system that he alluded to, I very much have to give credit to Erkin Saker for this. So this is a collaborative project between Erkin and myself, and Erkin is the one who asked the great question about neuroinflammation. And I would also point out, David, that you're going to want to work with a rat and not a mouse, because the neuroinflammatory response in the rat is much closer to the human than the mouse. So just got my 2 cents in on that. All right, so let's see, let me share my screen if I can. Let's see, it's telling me, yes, okay, there we go. What am I looking at here? For some reason, it's not, can you see my screen?
Yes, we can see the slides. If you wanna go into feature mode, yep.
Perfect.
That looks great.
All right. Okay, excellent. So somebody please let me know when I have 5 minutes left, because it was hard to get this down to 15 minutes. So what I'm going to be talking to you now is very different from what you just heard from David. And this is really looking at gene environment interactions that may be important in determining individual risks for Alzheimer's disease. So epidemiologic studies have really established a very strong association between air pollution, and particular traffic-related air pollution, which I'm going to refer to as TRAP, and near roadway exposures as risk factors for Alzheimer's disease, dementia, and possibly even age-related cognitive decline. So in order to really validate these epidemiologic data and begin to identify mechanisms by which TRAP influences AD risk, we're going to have preclinical animal models. However, if you look across the literature to date, most of the published animal studies have employed exposure paradigms that really don't capture the complexity and spatial temporal dynamics of real-world near roadway exposures. So they don't have the right composition, the dosing is usually way higher than humans would be exposed to, and the timing of the TRAP exposures are not really reflective of human exposures either. And we know that all these variables absolutely impact biological outcome. So our solution to this problem was to build an animal exposure facility next to a roadway tunnel and to house research animals in this facility and expose them to trap drawn directly from the tunnel into the exposure facility. So we chose the Kalakut Tunnel. It's a 1 kilometer long tunnel between Oakland and Orinda, California. About 60,000 cars per day pre-COVID would travel through this tunnel and through 4 bores. Each bore had 2 lanes. Another reason for choosing this particular tunnel, in addition to the fact that it's very heavily trafficked, relatively close to Davis, is that air quality has been very well characterized in this tunnel in prior exposures, or I'm sorry, prior studies. And so this is just a blueprint of the exposure facility that we constructed at the Kalakut Tunnel site. And so it basically has 3 sections. The middle section is the vivarium itself, where we house the rats. And we have an exposure chamber for the animals exposed to the tunnel air, and a separate exposure chamber for control animals that are exposed to clean air. There's also an instrumentation room. This is the engineer's side of the equation here. And so they put in all their instrumentation to really bring in the clean air and the tunnel air into the exposure chambers, but also to collect samples to be able to characterize those different air streams that are coming in. There's also, to the other side of the exposure chambers, a behavioral testing lab. So we actually did have some behavioral equipment that we set up for the studies I'm about to show you. So this is just a shot showing you what the vivarium looks like. So our engineers built these exposure chambers, and you can see that we can house 216 cages out at this tunnel facility. Half of the animals will be exposed to the filtered air, half of the animals to the tunnel air. This is an IACUC certified facility, and temperature, airflow, et cetera, is all controlled in this environment. So the first one we asked is, really, how realistic are these exposures to human exposures? And so this is work that was done by Keith Bine, who's one of the engineers involved in this particular study. And basically he collected air samples from both the filtered air exposure chambers. This is the air in the chamber itself, from the filtered air exposure chamber, as well as the trap exposure chambers, and then analyzed it for particulate mass concentration. He looked at both particulate mass of 2.5 size. So these are particles that are 2.5 microns in diameter or less. And this is a very important characteristic because this is a regulated particulate size. So the EPA, the World Health Organization, the California EPA, all regulate air quality based on PM 2.5 concentrations. This is also important because this is the size of the particle that is thought to have the most biological significance. These particles can get into our lungs and then be transferred throughout the rest of the body via the circulatory system. And there's evidence they can also access brain tissue. And so if you look at the data that are shown here, the filtered air is the sort of aqua color, the trap exposure chambers are this light gray. You can see that for any given day of the week, and this is an 18-month average, that we have significantly more PM 2.5 in the trap than we do in the filtered air chambers. And this is what we would expect. And it also shows you that the levels of PM 2.5 across these days remain very, very low in the filtered air exposure chambers. He also measured total suspended particles. This includes particulate matter of 10 microns or more in diameter. These are also regulated by EPA and other regulatory agencies. And again, you can see that if you compare the levels that are in the filtered air, which is the sort of darker blue color, versus the trap chambers, which are these darker gray colors, the levels are much higher. Now, the real important point to the slide is not only is there a clear difference between our filtered air controls and our trap exposures, but if we look at the annual guidelines for PM 2.5 established by the World Health Organization, we can see as 10 micrograms per meter cubed. And when we look at our levels, we can see that they're significantly below this annual guidance. So even though the levels of PM 2.5 in the trap are much higher than they are in the filtered air, they're still below the regulatory guideline. And this is a very important thing to remember as we move through the talk. So again, we're using trap levels that are within the World Health Organization regulatory guidelines, as well as the EPA limits. So the central hypothesis of our project that was funded by the ADRC, as well as we had an R21 from the NIEHS to fund this, was to study the question of whether or not chronic exposure to traffic-related air pollution or TRAP triggers inflammatory responses in the brain that initiate or accelerate the progression of AD. And I'm not going to go into what it depends on, I forgot to take that out, but we also looked at sex and genotype as factors modulating that response. So this is our experimental design. So you can see we have the two different exposures here, the trap and the filtered air. We looked at two different genotypes of rats, the transgenic F344AD rat. This rat overexpresses two human AD risk genes on a Fisher 344 background. So this is the APP Swede mutation as well as the PS1 delta E9 mutation. Both again are high familial AD risk genes. In addition, we also looked at their wild type F344 rat as a control for the genotype. And we looked at both sexes. So in all, we had 8 exposure groups. So that's the exposure groups. And then we transported these animals out to the exposure facility when they were about a month old. So at postnatal day 28. And then we allowed some of these animals to stay out there until they were 15 months old. But at 3, 6, and 10 months of age, we took some of the animals, a subset of the animals, and euthanized them to collect tissues. And then some of these, we also did behavioral analyses. So in terms of the outcomes that we measured, we did do behavioral testing. I'm not going to show you those data today, but I'm happy to answer questions about it. And we also took brain samples from these animals. Half of each brain was used for immunohistochemistry. The other half was used for beta amyloid ELISAs and p-tau westerns, as well as we did protein cytokine arrays by Luminex. And I won't show you those data today either. And so the outcomes we're looking at are plaque deposition, microglial morphology, CD68 immunoactivity, which is a measure of phagocytic microglia, reactive astrogliosis, and neuronal cell loss. So the first data I'm going to show you is something that was pretty startling to us. We actually were able to use spectral imaging microscopy to look for refractive particles in the brains of trap-exposed animals. So refractive particles would potentially represent these PM2.5 particles. What I'm showing you are images from the spectral imaging microscopy. We have the wild type brains represented in the top row, the brains from the TGF344 transgenic animals in the bottom row. And the left side of each of these is the filtered air animals. On the right side, the trap exposed animals. And what you can clearly see is in both the wild type and the transgenic animals, we see more of these refractive particles, which are these little bright dots, in the trap exposed animals relative to the genotype matched filtered air control. It's also notable that there appear to be many more particles in the brains of the transgenic animals exposed to trap relative to the wild type animals exposed to trap. Indeed, if we quantify that, that's what we see. So here we're looking at the number of particles per area in this case in the hippocampus, and at 3, 6, 10, and 15 months of age in these animals. The really important part, I think, is that the wild type filtered air animal along the bottom here, the open circles, we really don't see very many particles in the brains of these animals until they're about 15 months of age. But if we compare those to the transgenic animals exposed to trap, there's a huge increase in the particles in the trap exposed animals. Interestingly, the wild type trap exposed also show an increase in particle number compared to the filtered air controls, but there appears to be a very different sort of profile in the hippocampus of the wild type animals compared to the transgenic animals exposed to trap. Why there are more particles in the transgenic animal, we really don't know, but we suspect it might be due to the fact that the blood-brain barrier is likely impaired in these animals compared to the wild type. The other very interesting thing is if we look at the mean particle diameter, we see no difference across all the groups. However, we look at the mean maximum intensity, and this refers to the spectral intensity of these particles, we see a considerable difference between the filtered air and trap exposed animals in both the wild type and the transgenic F344AD. Our interpretation of these data is that it means that the composition, so the atomic composition of the particles in the trap exposed animals is very different from that in the filtered air exposed animals. We're currently trying to track down what the composition of this is, and our initial data suggests that it might be metals that are coming from catalytic converters.
Dr. Lang, you have just a few minutes left.
Okay, so let's move this quite along. So if we look here at, this is probably the part that Charlie is very interested in, is if we look at the effects of trap on plaque burden, we can see that in the filter air animals, we have a time-dependent increase or age-dependent increase in plaque deposition. If we compare it to the same age in the trap-exposed animals, we can see that there are more plaques present in the trap-exposed animals. So trap does increase the beta, amyloid beta plaque deposition. Interestingly, the female transgenic animals show plaques before the males, and this is accelerated by trap. And we didn't find any plaques at any age in the wild type animals. If we look at the ratio of A beta 42 to A beta 40, we can see again at 15 months that exposure to trap increases this ratio in the females and the males. And again, we're looking here at the transgenic animals. So two indices of Alzheimer's disease seem to be exacerbated by exposure to trap. If we look at tau phosphorylation in the male wild type rats, we can see that trap accelerates this as well. So we're looking here at an image from a filtered air animal versus a trap exposed animal, wild types. They've been stained for neurofilament. PHF1 is the antibody that we used. I'm sorry, this is for tau. And this is phosphorylated tau. This is the red. And then also for thio-S to look at amyloid deposition. We don't see any amyloid deposits in the wild type, but we do see an increase in tau phosphorylation in the trap exposed animals. In the transgenic animal, we do see these amyloid plaques. And again, we see phosphorylated tau associated with these, and this appears to be increased in the trap exposed animals. Indeed, if we start to look quantitatively at this, we see a significant increase in the wild type animals, males specifically, that are exposed to trap. This is quantified or confirmed, I should say, by Western blot analysis as well. Again, the key difference here is in the male wild type animals that were exposed to trap have a higher level compared to the filtered air controls. We also see an increase in neuronal cell loss in the trap exposed animals, and this is true in both transgenic and wild type animals. So if we look at the entorhinal cortex at 15 months, you can see fewer neurons in the wild type animals exposed to trap and fewer neurons in the transgenic animals exposed to trap compared to the filtered air controls. If we look at the CA1, we only see this difference in the transgenic animals. We didn't see any effects of trap on GFAP immunoreactivity, saw effects of age and genotype, but no effects of trap. However, we also did see very important effects on microglia. So trap did seem to increase the percentage of IBA1 positive cells that were also co-expressing CD68, which is a marker of phagocytic microglia, and that's shown down here in the yellow. When we quantify that, we can see that in male transgenic rats have an increased number of phagocytic microglia compared to wild type at any given age, and that's represented here by the pound signs. And if we look then at the effects of trap, we can see that trap increases phagocytic microglia in both the transgenic and wild type animals, and that's represented here by the asterisks. So we see this at 3 months in both genotypes. We see at 6 months in the transgenic animals, at 10 months in the transgenic animals, and in both genotypes at 15 months. Very different pattern in females, not so much with respect to just the filtered air controls. We do see again that transgenic animals have increased numbers of phagocytic microglia compared to wild type. But if we look at the effects of trap, we can see that increases CD68 positive IBA1 cells at 3 months in both the wild type and the trap, and at 6 months in both the wild type and the transgenic, I should say. But at 10 months, we actually see a decrease in phagocytic microglia in trap-exposed transgenic animals and a decrease in trap-exposed wild type animals at 15 months. So a slightly different pattern. So our overall conclusions are that trap exposure accelerates progression of AD neuropathology in the transgenic rats. It increases neuronal cell loss in wild type and transgenic AD rats. It increases microglial activation in both the wild type and transgenic rats, and that it modulates neuroimmune responses differently in female and male animals. And with that, I'll just end with the acknowledgments, and I'm happy to take any questions.
Thank you so much, Dr. Line. In the interest of time, I think we'll skip ahead, but if you don't mind staying on for a few minutes, if people have questions,
sure,
they might be able to ask in the chat. Thank you.
I'll also add that Dr. Olson, if you stay afterwards, we can ask the one question I missed in the chat for you later on.
Sure, no problem.
All right. I guess I'm going to take over now. So hi, everyone. It's been a great morning. I am Wayne Meyer. I'm an assistant professor in the Department of Neurology, and I lead the research education component at our Alzheimer's Disease Center, along with Dr. Carli. And I want to welcome you to our lightning round, where we will have exciting fast pitches from 4 of our REC scholars. And I'll go ahead and ask Dr. Audrey Fan to start sharing her screen, but each of our presenters will have 4 minutes to present on their research, and we'll have 1 minute for questions. So feel free to type them into the Q&A. And without further ado, I'd like to introduce Dr. Audrey Fan, who has done a terrific job managing this symposium. She is an assistant professor in the Department of Neurology and Biomedical Engineering. She is also the co-director of our Imaging Core at our Alzheimer's Disease Center. So Dr. Fan.
Thank you, Wayne. And just when we thought it couldn't be any faster, this is the lightning round, but I want to share our new lab and the MRI developments for non-invasive imaging in human, specifically of brain physiology and vasculature, that I think has a lot of future potential in terms of new methods for Alzheimer's disease. So I come from a stroke background. In a past life, I have strong training in MR physics, image reconstruction, and some of the methods that I've been working a long time on are non-invasive imaging of things like brain oxygenation. So it's something that if you think about MR and structure, it isn't necessarily clear that you can even measure oxygenation quantitatively, but with advanced acquisitions and reconstructions that target, for instance, deoxyhemoglobin in veins, like I'm showing here, you can actually reconstruct that magnetic susceptibility effect in a big magnet and quantify oxygen extraction fraction, which is a key biomarker of tissue health. And as I mentioned, I'm from a stroke perspective, so we've demonstrated this in acute stroke settings where in this graph, you're seeing a decrease of cerebral blood flow close to the lesion. And as you get away, the cerebral blood flow recovers toward normal levels. And with the measurements I was making using non-invasive MR, you can see a concomitant high oxygen extraction fraction close to the lesion, suggesting a penumbra of sorts. So this tissue around the lesion that could potentially be affected, it's trying to compensate by extracting more oxygen, but it's vulnerable, right? So this is the sort of pathology that I'm trying to assess physiologically with imaging. And a couple projects that are going on in my lab now, one is spearheaded by my awesome junior specialist, Corinne Donnet. And this really aligns with some of the Neuroimaging Core and Dr. Carli's grant in white matter hyperintensities, because clinically you see a lot of hyperintensities show up in older participants, but even though they're of presumed vascular nature, there still needs to be a lot of characterization as to what that vascular nature is and how is it different between people, especially in diverse backgrounds. So we're using something that is familiar from the Keynote, but arterial spin labeling to study brain perfusion. We're focusing on these white matter hyperintensities in the Adrc cohort and looking at layers around it. And one thing that Corinne has shown, which is really exciting, is that if you look at the center of the lesion, there is a reduction in cerebral blood flow. So that's far left here, this red curve, it's a reduction. But if you compare that to the gray, which is white matter, that's a deficit in blood flow that shows up even in the corresponding layers. So even if it's not inside the hyperintensity, there's that penumbra similar to the stroke concept that's showing up, and you see a similar destruction or microstructure dysfunction in diffusion concomitantly around these white matter hyperintensities. So we're using imaging to start characterizing these hyperintensities more. And the second project, I'll just quickly talk about my other awesome grad student, Greg Wheeler. He's developing a way to push these methods even further with a method called fingerprinting. So in this concept, if you have an MR method acquisition, for instance, you get multiple echo times with spin and gradient echo weighting. If you have a biophysical model of underlying parameters, you can simulate that and generate this dictionary. From a dictionary match, you are able to reconstruct the underlying physiology, and we're trying to do this for different microvascular pathologies. So in summary, we're very interested in brain physiology. We're trying to do this quantitatively with MR, and that is the focus of our method development lab. Thank you.
Great, thank you so much, Audrey. Are there any questions that we have? If not, we may just have all of the lightning round presenters talk, and then we can have questions at the end. So next in our lineup is Dr. Christian George. Dr. George, you can start sharing your slides. Dr. George is a postdoctoral fellow in the California Dementia Epidemiology Training Program, and she is part of Dr. Rachel Whitmer's lab here at UC Davis.
Thank you. So my research focuses on racial ethnic disparities and cardiovascular contributions to dementia. And so cardiovascular risk factors have been associated with dementia, and because they're often modifiable, are a great target for preventing dementia and also addressing racial ethnic disparities. Because we see that the prevalence of cardiovascular risk factors in Americans by race mirrors kind of the different racial ethnic differences we see in incident dementia. So on the left, you see that hypertension and obesity are most prevalent in Black Americans, followed by Latinos, whites, and then Asian Americans. Diabetes, Latinos have the highest prevalence, closely followed by Black Americans, and then Asian Americans and white Americans have the lowest prevalence. And then high cholesterol, white Americans have the highest prevalence, but it's pretty similar across racial ethnic groups. And so kind of mirroring that, the age-adjusted incidence of dementia is highest among Black Americans, then Latinos, whites, and then Asian Americans. And so some of my work has looked at early adulthood cardiovascular risk factors and neuroimaging using a racially and ethnically diverse cohort and images from MRI and amyloid PET. And so we looked at the prevalence of early adulthood risk factors when participants had a mean age of 32, and the prevalence of hypertension and high cholesterol was 37% in this cohort, 23% for overweight and obesity, and 5% for diabetes. And so over 40 years later, when participants had their neuroimaging done at a mean age of 75, we saw that the prevalence of amyloid positivity was highest in white participants, almost half, followed by Latinos, Black, and Asian participants. And then Black participants had the highest white matter hyperintensity volume, while Latinos had the lowest. And so we regressed these cardiovascular risk factors on the neuroimaging outcomes and found that early adulthood overweight obesity was associated with significantly smaller late life hippocampal volume, and early adulthood hypertension and hyperlipidemia were associated with significantly smaller cerebral volume. However, we did not see any associations between early adulthood cardiovascular risk factors and pathologies of amyloid burden or white matter hyperintensities. And so I've also looked at the prevalence of cardiovascular risk factors in Black Americans at different life stages with late life cognition. And so this figure shows the prevalence by life stage. You can see that overweight, obesity, hypertension, and hyperlipidemia were most prevalent in midlife adults ages 35 to 56, followed by young adults ages 21 to 34, and then adolescents had the lowest prevalence when they were ages 12 to 20. And diabetes had a pretty low prevalence, so we didn't look by life stage. But then we regressed these cardiovascular risk factors on late life cognition and found that hypertension, diabetes, or having 2 or more cardiovascular risk factors were associated with significantly worse cognition over 40 years later. And we did not find any statistically significant cardiovascular risk factor by age group interactions, which indicates that regardless of when these risk factors develop, they're detrimental to cognition. So we need to think about more of a life course approach to preventing dementia and Alzheimer's disease in Black Americans.
Thank you so much, Dr. George. That was terrific. So our last speaker for the day is Dr. Alyssa Weekley, who is an assistant clinical professor in the Department of Neurology. And so I'll let you take it away, Dr. Weekley.
Great. Can you all see my slides?
Yes.
Wonderful. All right, so I'm going to be talking about reducing caregiving and emotional divide despite physical distance between caregivers and care receivers. So as we all know, cognitive impairment results in a decreased ability to complete many everyday activities. But many older adults want to remain in their homes and be independent as they age. However, there is a lack of availability of in-home professional providers to assist with these everyday activities. And oftentimes the resistance among individuals that need some additional help can be high. As such, family members often assume caregiving roles. So another barrier is that there's an increased globalization. And so family members are often spread across distance. And even if they do live within similar areas, there is often a desire to maintain separate households. So this results in both remote and long distance caregiving. On a personal note, this happened in my family. My grandmother was recently diagnosed with Alzheimer's disease, and she's living in Palm Desert, California, but our family's spread all across the coast of Washington. We wanted to assist her in maintaining as much independence for as long as possible. But what I found was that there is a lack of evidence-based interventions that meet the needs of both the family caregiver as well as the individual with cognitive impairment. In addition to the lack of evidence-based interventions, physical distance can lead to emotional distance. And we can see loneliness and depression in the care receiver or the individual with cognitive impairment, as well as a sense of helplessness, worry, and burden in the caregiver. So a solution that I've been developing with computer scientists colleagues is the interactive care platform. We think that a technology-enabled remote caregiving platform will help directly assess the needs of both the care receiver and the caregiver, as well as addressing the emotional divide as well. iCare is an extension of evidence-based cognitive rehabilitation methods, and it enhances the original functionality of these methods, as well as accessibility using a web-based platform that is accessible from any device. So we really want it to be something that can be integrated into a person's existing life and what they're already using. So this here is the interactive care beta that we've developed. There are 5 distinct pages or areas that individuals can interact with on iCare. The first is a home page where the individual can see upcoming events and recent messages between themselves and the caregiver. There's a calendar where both the caregiver and care receiver can manage events and upcoming appointments. On the tasks page, you can see a breakdown of what's going on in that given day, as well as a to-do list. And the to-do list can be set both with and without a due date, and will remain on that page until it's completed. We also have a brain health center where individuals can both set brain health goals, such as physical exercise or cognitive engagement, and track them over time. And the care receiver can do this as well. So it can provide a sense of accountability and maybe a little bit of healthy competition as well. On the caregiver side, there's also the ability to set caregiver goals as well, such as number of contacts that they would make with the care receiver. Most importantly is the messaging center, where individuals can make video calls, they can send and receive pictures, audio messages, as well as existing documents. eye care is based on user-centered interface design, and we're doing 3 rounds of test and development with experts, caregivers, and individuals with cognitive impairment. Round 1 has been completed, and we've did interviews with experts. Overall, eye care was rated as a useful tool for both individuals with mild cognitive impairment and mild dementia, as well as their remote caregivers. Recommendations include minor modifications, enhancing some of the functionality, and expanding eye care to include sensors and other automatic detection devices, which we are now starting to complete in a 2nd round of development with eye care. I want to thank the Healthy Aging in a Digital World for providing funding for this pilot project, as well as members of the Everyday Cognition Resilient Aging Lab, our computer science team, and the expert panel that helped with some of these preliminary results.
Great, thank you so much. That was really exciting. That was a really fun round of great presentations from our early career researchers. Are there any questions, any final questions for any of our lightning round presenters or any of our speakers?
Feel free to go ahead and raise your hand if you want to. We're monitoring the hand raising. There are a couple questions previously, and I am navigating over to doing that. For Dr. Line, Hong Zheng asks, Dr. Line, thanks for presenting a fascinating talk, and she wonders if exposure to TRAP is removed in a given period of time, would you expect the outcome measures to be different between wild type and transgenic animals? Would these changes, in other words, be reversible or somehow permanent in their effect?
That's a really important and interesting question to which we have no data to address. So this is actually one of the questions that we hope to address with an R01 that we were just funded, based in part on the pilot funding that we received from the ADRC. So we will be addressing that question directly. But yeah, right now there really is not a lot of data to provide any guidance on how to answer that question. The one thing I will say is the USC Center that is headed up in part by JC Chen, who's done a lot of epidemiologic studies looking at TRAP and its effects on Alzheimer's disease in humans, has seemed to indicate that exposure during middle life, so during the middle ages of our lives, human life, are really the most potentially damaging in the context of TRAP effects on risk for dementia and AD. So there is a little bit of literature out there in the human side, but nobody's really tackled it yet on the animal side.
Great.
Yeah, Pam, that's exactly right. And I think that what's so beautiful about what you're doing is you are bridging that gap, because we had these kind of, may I say, coarse observations about cognition and brain related to exposure, but there was no a priori hypothesis why that might happen. Lots of things presented, and I think your work is really going to conclude with some basic mechanisms.
Thank you.
Other questions for some of our junior panelists would be great. Dr. Olson, Angela Zivkovic asks, I noticed you said that your compounds that you're synthesizing are lipophilic. I'm curious to know if or how they are transported in blood. Are they bound to albumin or lipoproteins, she asked?
Yeah, they're not super lipophilic. They have a basic amine on them, so they actually have a really nice profile for a CNS therapeutic. They're relatively water soluble, but they're also greasy enough that they can readily cross the blood-brain barrier. And so we don't have any major issues with these being bound to plasma proteins or anything like that. And so the pharmacokinetics are actually quite good. If you think about the pharmacokinetics of psychedelics, those are kind of pre-optimized for getting into the brain. And so the small modifications that we made for our non-hallucinogenic compounds really doesn't disturb those physical properties. And so they're still very CNS drug-like.
Oh, that's exciting. I did have the one question for you, Dr. Olson. Had you completed the pharmacological profiles for the receptor sensitivities? Are your novel compounds truly fairly specific for 5-HT2A, or are there other receptors? Is it a broad swath of either serotonergic or catecholaminergic receptors that they're affecting?
Yeah, so the two that, so I have to be careful because, you know, so the company Delix, they have hundreds of compounds now with very distinct, you know, polypharmacology. The compounds that I presented today, TBG and AAZ, they're the probes that my academic lab uses mostly. They are fairly specific. They only hit a couple other receptors, and we've profiled about 100 typical neuroreceptors. They don't hit cannabinoids. They don't hit dopaminergic receptors. They don't hit, you know, opioid receptors. The primary target seems to be the 5-HT2 family, 2A, 2B, 2C. 2B is not really in the brain, so it's not super important. And they also hit things like the serotonin transporter and monoamine oxidase, which is to be expected based on the structures.
Thank you.
This is Charlie. I just wanted a quick question for Dr. Sell. I really like your early results. They're fantastic. I know you didn't have much time to talk about why the MHC would be directly involved in synapse removal or modeling. And can you just give a second to tell us how that might work? Is this an autoimmune kind of thing? I don't think so. I think it's probably a normal physiological process. But can you share that with us?
So I don't have a lot of data about the exact mechanism that's happening in response to A-beta, but the lab, Dr. McAllister's lab, has shown that one of the mechanisms that it restricts synapse through is through MEF signaling, which leads to you know, changes in MEF signaling that lead to a restriction of synapses, and it's expressed in the postsynapse, which could lead to a synapse restricting environment. So my pet hypothesis is more that with an increase in MHC at the surface in synapses, that all of the normal like retraction and remaking of synapses in response to activity just makes it more likely that you'll lose synapses and then not remake them. But I'm working on the data.
Excellent. Very interesting.
Are there any final questions? I know we're 15 minutes over, but it's just been a really great discussion. Any last questions? Awesome. Well, with that, I want to thank Dr. Meyer and Dr. Johnson, as well as all the speakers for making this such a great experience, despite being virtual. I hope everyone got something out of the symposium. We're very excited to pursue collaborative research with the ADRC and your interests. We will ask the panelists in terms of being able to share the video recording, and once we solicit permission from everyone, then we'll hopefully follow up with a link. But in the meantime, if you don't mind, please filling out the link that Dr. Meyer sent. That'd be wonderful to get feedback and continue improving our symposium for the next year. And finally, as Charlie alluded to, it's great to have you present. If you want to apply for one of the research development projects, we would love to have you pitch your ideas to our weekly ADRC research meeting. And that's on Thursdays. If you can reach out to any of us, including Dr. Carli's admin, Shelly Aloko, we can schedule that for you. All right, thank you for joining. It's been a great morning, and I hope you have a great rest of the day. Hi, everyone.
Thanks, Dr. Fan. Good hard work.
Yes, great job. Thank you.
Thank you.
Thanks, everybody, for participating.